So in this section, we're going to talk about two of the more widely used biotechnological techniques. These are PCR and DNA fingerprinting. So whenever, crime investigators started looking at DNA, the major problem with any kind of DNA sample that they would pick up at a crime scene was that there really wasn't enough DNA to do any kind of analysis with it. I mean, you maybe had a handful of molecules of DNA, and you really couldn't see any of that with the current techniques. So PCR was actually a huge deal when it first came out, and still is today, because it actually hijacks the cell's natural way of copying DNA and does it just outside of the cell. So PCR, which is the polymerase chain reaction, uses polymerases, which are enzymes that basically will combine DNA nucleotides together, piece them together. So it uses these polymerases to copy DNA segments. So say this is your target sequence of DNA right here, okay? The PCR, polymerase chain reaction, makes many copies of that specific DNA sequence in just a few hours. Okay, either, like before the PCR, either that wasn't going to be possible at all, or it would take much, much longer than just a couple hours. It does this, through this process of PCR, it does what's known as amplification. So amplification is taking that small amount of DNA that maybe you collected from a crime scene and creating a whole ton of different copies of it so that you take that small amount of DNA and transform it into a large, useful amount of DNA. So PCR, again, uses the same theory as DNA replication, and it was through studying DNA replication that scientists were able to actually create the process of PCR. So PCR needs four things. It needs the DNA that it's going to copy, okay? So if you were going to copy a page out of a book, you would actually need the page out of the book in order to copy it. It's the template that's actually going to be copied. You also need DNA polymerase. So in this case, the polymerase, say, would be the copier machine. Okay, it's the mechanism through which you're actually going to make the copy. And then you also need your nucleotides. Okay, so with the whole book analogy, the nucleotides would be your ink. It's what you're writing the copy in. And then you also need two primers. Okay, so this is where that analogy kind of ends, because I don't really think about primers much. But this marks where the PCR needs to start on the DNA and where it needs to end. So it's kind of a bookend on both sides. It basically says start here and then don't go past this point. Right? This is all we need. So those are the four things that PCR needs. If you put all of those in a solution together and you go through the correct heating sequences, you can actually vastly increase the number of DNA molecules in your sample. Okay? So this is showing kind of the basic process. So you have your DNA strands, okay? First thing is they have to be kind of broken apart, unzippered in a way. And once that happens, then the primers will come in and find the sequence where they need to bind in order to mark the end points of the sequence of DNA you want to copy. Okay, so once that happens, then the polymerases will come in and start adding on the nucleotides. Okay, and that's how it all gets copied. And then it just continues going through cycles of this. So the three steps of PCR occur repeatedly in a cycle. And really this is controlled by heat. Okay, there are specialized PCR machines that you put your samples in and it does it for you. But you do have to set your parameters and things like that. So heat is used to separate the double-stranded DNA molecules. The cells actually have another enzyme for this. It's called helicase. But don't really want to put too many enzymes into your solution because it could start to degrade your compounds. And if you have helicase in there without its control mechanisms, then things get a little funky. So generally heat is used to separate the DNA strands. Then you're going to have the primers bind to each DNA strand on the opposite ends of the segments, okay? And this happens whenever the solution is cooled again. So you heat it to separate the DNA strands, cool it so that the primers can bind, and then allow the polymerase to work. And the polymerase generally has an optimum temperature, the temperature at which it runs the best, it works the best. Okay, so this is the process that we were just looking at on the previous slide. So when you go through a single cycle of PCR, you're doubling the number of DNA molecules. Okay, so with each cycle of PCR, doubling it, okay, so after 30 cycles, even if you just started with 1 molecule of DNA, you have over 1 billion copies of that piece of DNA in just 30 cycles. Okay, so that's maybe a couple hours. And when you have 1 billion copies of that DNA, you can actually run it on a gel electrophoresis and see the fragments of the DNA. Okay, there's enough there that you can actually see it. Versus if you ran it with 1, I mean, you wouldn't see anything, and there would be hardly any way to detect that. Alright, so now we start talking about DNA fingerprinting. So this is one that you've probably heard if you watch crime shows of any kind. Okay, so DNA fingerprinting is based on the fact that every human has certain areas of their DNA that are just repeated sequences. Okay, and it's not just that they're repeated sequences, it's that they repeat a certain number of times. Okay, so by basically counting the number of times those segments are repeated and doing that for a couple different segments that are similar to this, you can actually, with complete certainty, identify a single person by their DNA. And you don't actually have to look at all that much of their DNA in order to be able to identify them. So DNA fingerprints are based on these parts of the individual's DNA that can be used for identification. So these are usually non-coding regions, so they're relatively inactive regions of DNA. So they're not constantly being used to create proteins, okay? And it really focuses on the repeating DNA sequences. Sorry, Potter apparently is not very impressed. But the number of repeats will differ between different people. And so on those crime scene shows, when they start talking about 13 alleles in common and yada, 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 that is, yeah, not exactly what they're looking at. They are looking at these repeats, okay? And then because if you have more repeats, you're going to get a longer piece of DNA, and longer piece of DNA means it's going to move a shorter distance through a gel electrophoresis. So when you run everything in a gel, you get this banding pattern, okay? So that is very similar to what we looked at in the last chapter with the different bars on it. So, example of a DNA fingerprint, okay? So if we look at this sequence here, say this is the sequence of the repeat, okay? So it's 33 base pairs, and it can be repeated multiple times in a sample of a single person's DNA. Okay, so we look at the DNA segments from person A. They have 4 repeats, then 3 repeats, then 6, then 7. Okay, so 7 will be at the top because it is the largest. Then you'll have 6. Underneath that, you'll have 4 and then 3. Okay. Now, person B, they just have 2 repeats, 5, 3, and 4. So you're going to have 5 at the top, okay? Then you're going to have 4, right, which will match up with the number of repeats for person A here. So these will be in line with each other. And then you'll have 3 repeats, which again lines up with the ones from person A, and then 2. Okay, so you can obviously tell, even if you mixed up these DNA samples, that they do come from 2 different people. Okay, because looking at the same regions of DNA, you have different numbers of repeats. So DNA fingerprinting can be used for identification. So the crime scene shows do at least get that part right. So this is an example of a DNA fingerprint. Now this is not just used for matching someone's DNA, like if they left their DNA at the scene of a crime or something, it's not just used for that. It's also used for paternity tests, right? Which we'll get into in a little bit. But you can see here you've got the mother, 2 children, and the father. So because the 2 children only got their DNA from either the mother or the father, every band that you see in the kids has to have a matching band in one of the parents. Okay, it doesn't have to be both of the parents, but it has to be from one of the parents at least. Okay, you see here this top band matches across from the mother, and she gave it to both kids. Then you see the father and the mother have a similar band right here, passed on to one kid. The father then passed on this band to both kids. Mother and father also have similar bands here, okay? And then so on and so forth, just going all the way down, right? So like for an example here, we've got the top part of this band comes from the father, bottom part is from the mother. You can see the markings that they did to kind of analyze this DNA fingerprint. So DNA fingerprinting depends on the probability of a match. So we're going to look at the probability. You don't have to calculate this. Okay. So many people have the same number of repeats in a single region of DNA. So like we saw in the previous slide, we had 2 different people and they had 4 repeat segments and 3 repeat segments. Okay. But what you'll notice is that the other 2 repeats that we looked at were not identical between the 2 people. Okay? So the probability that 2 people share identical number of repeats in several locations is negligible. In fact, in order to prove identity in a court case, they actually have to get it so that the probability is basically only 1 person in everyone who lives on Earth. Okay? So 1 person in 7 or 8 billion, right? Because usually they are fine overshooting that just to make sure that the probability works out. So individual probabilities are actually multiplied together to find the overall probability of 2 DNA fingerprints randomly matching. Okay, now of course this does not account for identical twins or anything like that because that's kind of impossible to account for in this. But, okay, so say 1 in 500 people have, say 6 repeats at this one location. 1 in 90 maybe have 5 repeats at another section. And then 1 in 20 might have 13 repeats at another section. So basically this is looking at, okay, so you're the 1 person in 500 in this location, and you're 1 person in 90 in this second location, and you're 1 person in 120 in this other location. So you multiply these probabilities together, and you get 1 chance in 5.4 million people that you will have a duplicate of those 3 conditions. Okay, so it's all looking at the probability of this. So several regions of DNA are used to make DNA fingerprints. And really they use as many segments as they need to prove that it's only 1 person on Earth can have that combination. So we're going to pause here and pick up in the next video.